and good morning everyone for those that are in Europe and about the others depending on where you're standing now. So here we have today connected myself, Manuel Mameli from Lausanne, but especially Bart the Strooper, our keynote speaker of this morning. So Bart, thanks a lot for connecting at this early morning hour. And so we have Bart here. Why we have Bart sitting with us today? Because he's going to tell us about the secrets of Alzheimer's disease in a sort of way. Bart is an expert in the field, studying neurodegeneration in general, more specifically for the context of today, Alzheimer's disease in his intracellular form. How does that happen? The trigger motion is career has been devoted indeed to analyze what are the pathways that may lead to the disease, and especially the disease in humans by using and comparing, paralleling with animal models. So without further ado, I will let the stage to Bart for this talk. I invite all of you to ask a question through the QA answer, and we are going to deal with it, I would say, at the end of the talk, once Bart has told us everything. So the show is open and the stage is yours. Thanks, Bart.
Thank you very much, Manuel. So good morning to everybody. This first slide summarizes basically the raising insights into Alzheimer's disease. And so as you can see, I have this indicated this biochemical phase, which a lot of focus has been over the last years, over the last decades, in fact, because these are the typical biochemical lesions you see in the brain of patients when you look in the microscope. And these are abnormal accumulations of the A-beta peptide, this amyloid plaques, and then abnormal accumulations of the tau protein, which are these neuronal tangles. And so a lot of work has gone through this. The other side of the work has been on the clinical representation of Alzheimer's disease. And so, as you all know, Alzheimer's disease presents as dementia. If you look in the brain, you can see brain atrophy and a couple of lesions. And nowadays, we are also able to visualize these biochemical lesions in the brain. So basically, we can define Alzheimer's disease as a dementia with presence of amyloid beta and abnormal tau. The big oversimplification in this is that it takes actually more than 20 years to come from these biochemical lesions, the first appearance of these biochemical lesions in the brain, and the clinical phenotype dementia. And the second big problem here is that dementia is a symptom from many other diseases, other neurodegenerative diseases. So it's a symptom and it's not really sufficient to talk about Alzheimer's disease. So over the last 4 or 5 years, we have started to look much more about what links these biochemical lesions to the brain atrophy in the late stage of the disorder. And the reason is obvious. If you can work on this intermediary phase, you will probably prevent dementia. And so the cellular phase is characterized by changes in all the cells of the brain. So today I will talk about the insights we have in this cellular phase. So a second thing I absolutely need to clarify is that even sporadic Alzheimer's disease, so the large bulk of Alzheimer's disease, has still a very strong genetic component. So ApoE4, apolipoprotein 4, is the strongest risk factor. But Swedish studies in twins have shown that about 2/3 of the risk of Alzheimer's disease is probably linked to your genetic makeup. And so this genetic makeup has been partially characterized in genome-wide association studies. And here you see the classical Manhattan plot. And you see here at least 40 genes now, which are canonical genes or gene loci, to be more precise, which have been associated at genome-wide significance with Alzheimer's disease. But below this threshold, there are also many genes, as you can see here, which show a certain association, a certain statistical certainty to be associated with Alzheimer's disease. And these many other genes add to, when you calculate or when you add them to a polygenic risk score for Alzheimer's disease, they add to the risk for Alzheimer's disease. So showing that also these genes, which are the gene loci, which are below the threshold, are adding to your risk of Alzheimer's disease. And so this makes it very feasible to ask a couple of important questions. So the first is, of course, how do we use this genetic information to generate a functional hypothesis for Alzheimer's disease? The second point, which is very important, is to ask the question, how and under which conditions will genetic risk manifest? And it's important that you realize that these gene loci, which have been identified, are risk loci. So that means that they will only manifest when you have risk. You can compare it with driving a car with a belt or without a belt. When you drive normally and you have no belt, you will not really know, or you don't see the impact of not carrying this belt. But when you have an accident, then the belt becomes important. And it's the same with genetic risk. It's only when you test it in a situation where the risk is important, you will see what its role is in the disease process. And that makes it, of course, very difficult to understand these genetic risk factors in research. And then finally, so this is the essential question then, can we investigate these risk genes in a pathological relevant condition? And so the problem is that you need a good model to do that. And Alzheimer's disease has no good animal models. We have mouse models which develop these amyloid plaques or these neuronal tangles, but none of these models have a convincing link between the amyloid plaques and the neuronal tangles. And they also have no convincing link towards neurodegeneration and dementia. So some aspects of the disease are recapitulated in these two different models, but there is no complete model. On the other hand, we can use brain material from patients suffering from Alzheimer's disease, but that's really the late stage, that's post-mortem. There are changes there which are caused by the agonic state of the patient, and we miss completely the initial phase of the disease, which is, in my opinion, the most important one to study. So we were thinking, can we bring together the human genetic data, which are clearly linking molecules to disease, with experiments coming from the mouse models, mouse transcriptomic data. And so in the next few slides, I will show you that we can. So here you see the setup of the experiment. So we took amyloid mouse, APP, we call it, amyloid plaque forming mouse, controls and transgenics, tau tangle forming mouse, controls and transgenics. We took the hippocampus at 2 stages, 4 months, which is initially of the in early phase of the disease in these models, and 10 months, which have behavioral changes. So the 2 mouse have similar behavioral changes. And so we can look at 4 months and at 10 months what happens, and we can compare the wild type with the transgenic at age, genotype, and we can also study the age and genotype interaction. I have no time to go into the technical details, but we can look to the final results, which are very instructive. So you have here the comparison over age between the tau model and the APP model. And you see here the differential expressed genes listed over this graph. And what you immediately see is that most of the genes cluster around this transversal axis, showing that changes in the tau and in the APP model over age are rather similar. So these 2 mouse models age in a similar way. When you compare the APP, the amyloid mouse, with the tangle mouse, and you compare and you do the same comparison, then you see that there are dramatically differences in the way that the tau affects the expression of genes and the APP or the amyloid plaques affect the expression of genes. And you see here, for instance, these yellow changes along the tau axis. These are genes which are mainly involved in neuronal function. So that's very interesting. Our mouse mainly downregulate genes involved in neuronal function. And this fits with the idea that the tau pathology is very close to the neuron pathology. For the amyloid and the cognitive changes in Alzheimer's disease. For the amyloid beta pathology, you see complete different patterns. So most of the genes are upregulated. So these are the downregulated genes, these are the upregulated genes. Most of the genes are upregulated, and they are a complete different bunch of genes. There are genes involved in inflammation, neuroinflammation. So the conclusion is that there's very little overlap in these 2 mouse models. So the tau changes neuronal genes, the amyloid changes inflammatory genes. And so when you look for the age genotype interaction, you see that there are little changes in the tau axis. So the changes are occurring immediately and affect neuronal function very early on, as soon as the tau transgene is active. But for the APP, we see that over time, many more genes start to be changing. So there is a time effect in the amyloid mouse. So now the whole challenge was here not to identify what happens in the mouse, but to see what happens in human and whether we can make sense out of the changes in the mouse and to what extent the changes in the mouse reflect the genetic risk of Alzheimer's disease. And to do this, we go back to the GWAS studies, which I was explaining to you, and where you see that at the genome-wide, well, slightly below, so at the genome-wide, which is 5^-8, we have 40 genes, should be listed here. And then if we increase or lower basically the threshold for the p-value, the cutoff, you see that we get increasingly more gene loci included in the analysis. So for instance, at 0.1, which is a high p-value, admittedly, you see that about 2,700 gene loci contribute to the overall risk of Alzheimer's disease. And so what we did here is take this list of Alzheimer's disease genes or loci and our age genotype and age genotype interaction, and we did gene set enrichment analysis. So we looked for the presence of these genes in the changed genes in our different comparisons. It's a complicated explanation, but we have published this. You can find that back in the paper of Annerieke Sirksma in Embo Molecular Medicine. But so to make a long story short, what we found is that there is only a strong enrichment of human genome-wide association gene candidates in the interaction of amyloid with age. So there you see that whatever threshold we take here, there is a strong enrichment of Alzheimer's disease risk genes. So this brings, so when we look then to these AD risk genes, they are all enriched for inflammatory functions. Not surprisingly, as I told you already, the genes which are changing in the amyloid mouse are mainly involved in inflammatory functions. So immune response, regulation of cytokine response, etc., etc. All these categories are enriched in the response in the mouse to the amyloid plaques. And so here you see an interesting intersection. So we take here all the genes which are identified in GWAS studies for Alzheimer's disease at the p-value of 0.001. We take here the interaction of all the genes which are differentially expressed in the APP transgenic amyloid plaque mouse. And then we take all the genes which respond to the amyloid APP blue module. And you see here that in the intersection of this, we find 18 genes, some of which have been identified as the canonic genes for Alzheimer's disease, so the high risk for Alzheimer's disease, including ApoE, clusterin, CD33, PLC gamma 2, which are very well-known genes associated with Alzheimer's in GWAS studies. But then we find also a couple of genes which at the same level of risk are identified in our approach and which were very low, with very low certainty associated with Alzheimer's disease in the GWAS studies. And these are this bunch of genes like a TREM2-like gene, GPC2, a SIG gene, XB, etc., etc. So this shows you that by this approach, we can enrich for interesting genes involved in Alzheimer's disease. And so to make this story, to wrap up this part of the story, so what we find is basically that the gene risk for Alzheimer's disease, that we find deregulated genes, which are in the loci associated with risk of Alzheimer's disease, they determine how microglia respond to the amyloid plaques. And here you see a couple of functions, again, repeated, as I said, and this is a representation of the microglia and all the different genes we identified in response to the amyloid plaques. So the conclusion of this story is basically that the genetic risk genes in sporadic Alzheimer's disease define an inflammatory response to amyloid plaques, much less to tangles, and point in the first place the microglia for the cellular response in Alzheimer's disease. Obviously, this is an oversimplification. I'm just saying that microglia are an important part of the cellular response. And as we will see later on, we have also data showing the involvement of the other cell types, as I indicated in my introduction. But let's now focus a little bit more on the microglia. And so we wanted to know what microglia are really doing in response to the amyloid plaques in our transgenic or in our knock-in mouse model for Alzheimer's disease. And again, to make a long story short, we took our mouse model, we look at 3 months, 6 months, 12 months, and 21 months, which gives you an increasing amyloid plaque deposition. And we isolated the microglia at the different stages to look what happens in the microglia response. And so here you see the classical cloud, the Disney plot of the microglia, the different. So every dot is a transcription profile of a microglia from those experiments, and they are all put together here. And you see that we get 6 large clusters of microglia, so different transcriptionally defined groups of microglia. So the most important one here is this amyloid response or activated response microglia. We have also these homeostatic clusters. But let's look to the data, to the essence of the data, which you can see summarized here. And so what you see here is these 6 different microglia clusters. And in brown are the 4 stages of the amyloid plaque forming mouse. And you see that this homeostatic cluster is relatively stable in the amyloid mouse, and you see the same. It's a little bit higher. There are more of homeostatic microglia in the control mouse. But then you see immediately here a strong decrease in this homeostatic cluster 2 in the amyloid plaque forming mouse over time, and that's shifted. These microglia are shifted to another profile, which is this activated response microglia, and you see that this increases very rapidly in the amyloid plaque mouse. You see the same change, but much less outspoken and only at a very late stage. 21 months is very old for a mouse. You see that also in the aging normal mouse, we see this activated response coming up at 21 months old. So this is very interesting. The amyloid plaque forming mouse use a response in the microglia, which you can see to a much less extent in aging microglia. And so we can then also do pseudo timing. So here you see all these microglia again, but now put in an order the most similar microglia closer to the beginning of this pseudo timing representation. And then you see that basically these microglia may evolve into very dark group and an activated response microglia group. And so to make, again, a long story short, you can then go for the gene expression in these different arms. And you see here a strong enrichment in the activated response microglia of ApoE. So ApoE becomes becomes more than 100-fold upregulated in microglia which are exposed to the amyloid plaques. And so, as I said already, ApoE is the major genetic risk factor for Alzheimer's disease. And this is really surprising to see that this major risk factor, which is usually expressed in astroglia, appears to become strongly upregulated in microglia when they see amyloid plaques. There are also other genes, but I have no time to go into the details. And so this is another representation to show the strong ApoE upregulation in the amyloid response genes. So where is this ApoE expressed? Here you see an in situ hybridization experiment with an amyloid plaque in the brain of our mouse. And you see the nuclei in blue. And then you see here the ApoE mRNA expressed in this microglia clustering around the amyloid plaques. And it's really only in the amyloid plaque exposed microglia that you see this upregulation of ApoE, as you can see here also. So this brings us to the question whether ApoE is really important for the microglia activation, or is just a marker of a cell state of the microglia. So we went for an ApoE knockout and repeated our single cell experiment as shown here. And again, to make a very long story short, here you see the different categories of the percent of microglia per genotype in these famous activated response cluster. And this activated response cluster is in fact very similar to the DAM cluster as defined previously by Ido Amit. So you see, if you take ApoE away, that so you see here this amyloid mouse has a lot of this ApoE positive or increased expression microglia, and you see that in the ApoE knockout, this cluster almost disappears. Another cluster, the immune response, the interferon response cluster, which is also increased in the amyloid mouse, is not affected by the ApoE deficiency. So it's very specific it's very specifically needed for the ARM or DAM response of the microglia in this mouse model for Alzheimer's disease. And so when we look again to the amyloid plaque here, now this time in white here also, and you see here, this time in red, the ApoE, but it's now the protein which is stained. So it's an immunohistochemical staining. The previous one was mRNA staining. And you see that this amyloid, this ApoE is secreted by the microglia and binds to the amyloid plaque. So it's really covering the amyloid plaques with ApoE. And here you see these activated microglia, which are swollen and which are associated with this amyloid plaque. And so in the ApoE knockout mouse, where we had no activated microglia anymore, first of all, you see this much less dense amyloid plaques, indicating that ApoE has an effect on this amyloid plaque formation. And then what you see also is that the microglia are present in this brain, but they are not forming these clusters of activated microglia around amyloid plaques. So this clearly shows that the major gene, the major risk factor for Alzheimer's disease, is involved in the response of the microglia to the amyloid plaques. And so the conclusion of this part of the story, and I could go in all the details, but is that ApoE, the GWAS risk factors of Alzheimer's disease, not all of them, but several of them, age, as I've shown you, and gender, for which I had no time, but the female microglia become much faster into this activated response mode than the male microglia. So ApoE, GWAS risk of Alzheimer's disease, age and gender affect microglia response to amyloid plaques. So this brings me to the next step in my presentation, and that is what basically happens now in this amyloid plaque niche. So I showed you, and this is a picture of Alois Alzheimer, and he was already obsessed by the cellular changes in this amyloid plaque niche, the cellular niche around the amyloid plaque, as you can see here. And here you see a picture I took from Michael Henneka, which shows this extreme inflammation of microglia around the amyloid plaque. But it's very clear that it's not only microglia which are responding to these amyloid plaques. And so what we wanted was an unbiased approach to what transcriptional changes occur close and far away from the amyloid plaques. And that's why we introduced a couple of years ago spatial transcriptomics in my lab. And just before I start with discussing those data, here you see again our mouse model represented at 3, 6, 12, and 18 months. And you see the accumulation of the amyloid plaques, which is really massive at 18 months. But when you look at microscopical level, you see here these amyloid plaques, and you see now not the microglia stained, but the astroglia. And so the astroglia also give a big response to these amyloid plaques, which is relatively mild at 3 months, but it becomes really massive at 18 months. And so we wanted to know what happens in the cellular niche of these amyloid plaques. And so here you see basically the setup of the experiment. We take a slice of 10 micrometer, which we stain. We have a slice here of 10 micrometer, which we use for the spatial transcriptomics. And you have another slice of 10 micrometer, which we again use for staining. And so these two slices give us morphological information of the transcriptional changes, which we will analyze in this slide. And so here you see a picture. This is a brain slice on top of such a spatial transcriptomic slice, which we did together with Joachim Lundenberg and his collaborators. And so what you do is you permeabilize this tissue, and then you have the mRNA will diffuse on these spots, and on these spots you have poly-T sequences and a barcode, which is of course extremely important for the final result. Then the mRNA is binding to these spots, and then you can afterwards release the cDNA, and thanks to the barcode, you know where the mRNA, which you have sequenced here, is coming from, and you can reconstitute this picture, but this time with the transcriptomic information all over the place. And so then you can do this type of Disney plots again. This time not every spot here is now a tissue domain or a cellular domain around an amyloid plaque, or a tissue domain from the same size. And you can do expression analysis. And again, just to show that this really works, you see that we can cluster, for instance, the hippocampal areas in different clusters of this, that is tissue domain can be clustered according to different hippocampal areas. And you can cluster them also according to disease, which gives you the other axis here, where you see, for instance, that the wild type of 3 months is here, that AD of 3 months is here, and that then and that the other groups also cluster below. So this shows you that our sequencing worked. And then we have a lot of information of every tissue domain. So we know where it is in the brain, we know whether there is a lot of astroglia, etc., etc. But we have also this plaque index. And so what we did is we developed a method to automatically assign a level of amyloid exposure to a tissue domain. So we use tissue domains with 3 different exposures of amyloid. And so we have a quantitative way to measure that. And here you see then again this Disney plot, and we overlay here the intensity of the amyloid plaque spots. And of course, you see here in the young mice, in the cluster of the young mice, non-amyloid mice, that there is almost no plaque staining. And here you see the plaque staining in the tissue domains of the older mice. And so now we can do very interesting, all kinds of analysis, again, just in a nutshell. So we have 10,000 transcriptomic profiles, 4 time points, wild type and transgenic. And we have a regional annotation, and we have a plaque index for every spot. So we can ask here, what are the molecular alterations in the plaque niches? We have a temporal resolution, we have a genotype difference, and we have a spatial resolution, close to plaques versus distance to plaques. So here you see a picture again. And so I will show you one graph with real results. And so the graph is as follows. So you have here the axis of plaque exposure. So we have data for every gene, to what extent it is exposed to plaques. We can then do a differential expression of transgenic versus wild type, so genotype in this axis. And so here you will see the genes which are strongly upregulated in the amyloid plaque niche. And here you will see genes, for instance, which are upregulated in non-amyloid plaque area. And this is according the genotype axis, and this is according the plaque axis. And so here you see a part of the data. They are really massively, and I invite everybody to mine the data, which are completely available on our website. So here you see a first group of genes in red, which are which turned out to be upregulated according to the amyloid plaque match niche very early at 3 months, when there are no other symptoms in the mouse. And so we think that this is a kind of protective response to the amyloid plaque, increasing amyloid plaque loads. And when we analyze those genes, they are all oligodendrocyte or myelin related genes. And very interesting, at 18 months, this cluster of genes is downregulated. And then on the other hand, we have the plaque-induced genes, PIC we call it. These are this purple group of genes, which are not affected in the beginning at 3 months, but which then becomes hugely upregulated, both according to the genotype and the amyloid plaque niche. And when we look to the identity of these genes, they are all related to microglia, astrocytes, and you can see the annotation here. And when we did an overlap with the arm phenotype, which I just described to you, there is a part overlapping with this activated microglia. There is also a part of the genes overlapping with the previous identified activated astrocyte phenotype. And then there are several genes which are more or less novel and which have not been associated to these genes in the past. And so here you see the list of the genes, no time to go in every gene, but overall, we see a strong enrichment in gene ontology for complement activation, endocytosis, lysosomal degradation, oxidative stress, immune response, and antigen processing and presentation, which is likely to overlap with the ARM response. So the most interesting and fascinating result is basically here summarized in this plot. So we have here all the ARM genes, all the big genes put around, and you see here how they interact with each other in a WGCNA analysis. So basically, we analyzed here how these genes change with each other together in different conditions. And then they automatically clustered in this green group of genes, which are mainly astrocyte genes. And you see here how they are co-regulated in the wild type situation. And then here a bunch of microglia genes, which are also weakly co-regulated in the wild type situation. And then these genes, which are not much co-regulated with neither the astroglia genes or the microglia genes. And then you see here for increasing exposure of amyloid. And then all of a sudden, these genes start to interact with each other. So I mean, they start to be co-regulated with each other. And so this suggests very strongly that the astroglia genes here and the microglia genes here are co-regulated over these two cell types. And also these genes, these extra new genes, become incorporated in this WGCNA network when exposed to high levels of amyloid plaques. So this brings me to the next conclusion. The PICs are a gene regulatory network over microglia, astroglia, and oligodendrocytes, coordinating the cellular response towards amyloid plaques. So I'm going to skip this conclusion slide because I mentioned all these things already while speaking. And so the last 5 minutes, I just want to share an initial model we are developing now to study and to translate these findings into real human microglia and in real human brains. I'm not going to talk about our neuron chimera mouse where we transplant human neurons derived from iPSC cells in a mouse brain and have very interesting phenotypes, but you can find some details about that work in our paper in Neuron. But I can tell you that we developed this model now to a very, very nice full model, but I have no time to explain that today. But I'm just going to briefly talk about the work of Mancuso, Renzo Mancuso in my lab, who developed a nice mouse model for human microglia. And one of the reasons that we did is that we realized that although the mouse model, as I've shown to you, mimics aspects of the human disease, a mouse brain is not the same as a human brain. And a lot of genes which are expressed in human microglia are not very well expressed in mouse microglia, as I will show you. So we developed a mouse model where we could transplant human microglia derived from iPSC cells into a mouse brain. And here you see a section of a mouse brain, and every red dot is a human microglia which has replaced the normal mouse microglia in this brain. So you see, it's a very effective method. And here you see characterization of these microglia. They are sitting in the same niches as the mouse microglia, as you can see here. So they really go and fit themselves in the mouse brain. And here you see then pictures of these microglia nested in the mouse brain. And so then, I mean, is this important? Well, I can show you here an experiment where we injected amyloid beta into the tail vein of a mouse transplanted with human microglia. And we tested the effect of this amyloid, which goes into the brain, on the expression profile of this human microglia and the mouse microglia. And here you see that this is real. Here again, Disney plots, no time to explain, but you see that the beta has massive effects. And here you see a temporal curve, again, such a pseudo timing. And here you see profiles of individual genes which are changed when the mouse and the human microglia are exposed to the amyloid. And you see really that in these clusters, which are chosen because they are overlapping or similar in the mouse and the human, but you see that in these clusters, these genes have dramatic different responses, especially here as you can see here in the human mouse, in the human microglia, the extremely different responses compared to the mouse microglia. So it's important to take the human component in the modeling of Alzheimer's disease. And so if we briefly compare the transcription profile of the mouse microglia, and we look to genes which are relevant for Alzheimer's disease, like APCA7 or BIN1 or other genes which have been identified in GWAS studies, SORAL1, for instance, then they are present and they are interesting to study in the mouse microglia. But when we use human microglia, you see a staggering increase in genes which are not really very well expressed or are very different in composition in the mouse microglia. And you see here really the relevance to use human microglia to study the genetic risk of Alzheimer's disease, in particular when you want to do gene interactions and try to understand how a polygenic risk score affects the behavior of this microglia. And so this work is underway, and I hope to talk about that in the next seminar. So the overall conclusions of my talk are that in sporadic Alzheimer's disease, amyloid plaque is a trigger, not a driver of the disease. So you can have plaques, but you don't need to become diseased if the cellular response becomes under control. And so the cellular response, I want to expand it from microglia to astroglia and other cells, is determining whether you get Alzheimer's disease or whether you are protected against the trigger of amyloid plaques. The neurodegeneration is probably downstream, or it's very likely downstream of the neuroinflammation. And neuroinflammation, you need to see broader than microglia. It's a response of the oligodendrocytes, astroglia, and microglia. And tau is downstream of the cellular interaction, as I tried to suggest in my first part of the talk. So the people involved in this work are Wei-Ting Chen, who has really been the driver of the spatial transcriptomic work in my lab, and then Mark Thierse and Ashley Liu, who have done most of the bioinformatics with a lot of clever tricks to analyze the spatial transcriptomic data. Carlo has pioneered the single cell analysis of the microglia work, which I've just shown to you, together with Nicolas Fattorelli, who was responsible for most of the bioinformatic analysis of those experiments. And then Nicolas works also together with Renzo to develop this microglia model I presented to you at the end of my talk. And then Amerike Sirksma, and there should be an R here, Sirksma, has been central in the first part of the talk where we compared mouse and human for their responses to amyloid plaques. And also she worked together with Ashley and Mark for the bioinformatics. So thank you very much. This is my final slide with acknowledgements for my different funding resources, and a drawing here of the UK Dementia Research Institute, which is a fantastic initiative in the UK, where we bring together centers in different top universities all over the UK to work on dementia, and where we hope to find cures for this devastating disorder. Thank you very much for your attention, and I will stop sharing my screen and see whether there are any questions.
Thank you, Bart.
Thank you, Manuel.
As usual, incredible amount of data, and I invite all of the attendees to share your question across the Q&A, and I will then lead it to Bart for the discussion. For the moment, people, I guess, are still writing, but then I have a couple of points maybe to launch already the discussion. Bart, according to you, okay, microglia seems to have, together with astroglia, a central role. But then, if I'm not mistaken, what you provide us here is a sort of a snapshot in different momentum that are going across the disease. And you call this activation of microglia. But here I wonder, how far or how close are we to understand the real temporal dynamic of these microglia? I mean, I guess these guys are going up and down, controlling very timely this. So what do you think we are still missing, and at which level of analysis?
We are missing a lot. We are missing a lot. I think the main thing here is that all of a sudden this simplistic view that amyloid is causing synaptic dysfunction and then you get a whole disease becomes a really complicated interaction of different cell types where you get protective and damaging responses. And I didn't have time to talk about that, but the main drive of my research is to understand why certain cellular responses will keep your brain going even if amyloid plaque is there, and what responses are going to lead to a decaying brain and a collapse of the brain. And so I didn't talk about the neurons and the neural circuitries, but in the end, it's all about how long you can keep your neural circuitries working normally. And so, the point I want to make here is that your microglia and astroglia are a very important aspect of the defense of your circuitry towards these accumulating proteins. And so that's basically it. And so the second point is here, the dynamics of the whole system. That's also why I think that we cannot go further without our mouse or animal models, because the only way to capture the process over time is using such models where you can start from the beginning, where patients, humans, have no signs, nothing. So you even don't know. So it's even making our studies, case control studies, complicated, because you probably also have in these control groups patients which have biological Alzheimer's disease without any clinical phenotype. So we cannot capture that very well in humans. So we need the models there. So that's an answer. A second answer. And then the third part of the answer is that indeed all these cells have very dynamic responses, and it's probably that they become that you have upregulation of this ARM response, but it would be then downregulated again at phases where the disease is less pronounced, or whether when there is something, it could be that an inflammation, peripheral inflammation, activates the whole thing and drive the disease a little bit more forward. So it's complicated interaction which goes over decades, then to finally end up with the collapsed brain. And then it's too late to treat also. I think that's a very important realization. We don't have to treat Alzheimer's disease. Well, we should try to do the best what we can for these people, but we will not have a good treatment at that stage. So that's also an important insight.
Okay, Bart, you have a question from Andreas Noll.
Yes.
I read the question for you maybe, so people get to know. Would it be correct to say that there is a transition between a first phase, which is potentially reversible, and in which amyloid puts stress of the tissue, or this triggers the disease, or a second phase that is rather tau-driven? How do you see it?
I agree with that type of thinking. And we know basically that this is the case from the familial Alzheimer's disease cases, which are all mutations which increase amyloid beta or change amyloid beta. And so they all end with dementia, and they all end with tangles and tau pathology. And the tangle and tau pathology is in these patients, again, more correlated with dementia than the amyloid plaque accumulation. So there is a dissociation between the clinical phenotype and the initial biochemistry of the disease. And so the model which I try to present fits much better with such a complicated, explains or potentially explains much better the disease even in the purely amyloid-driven pathology. And so the logic is indeed to think then, because it takes so much time, that you have a whole time where this amyloid is in your brain and irritates or stresses your brain, but your brain is just able to cope with it. The microglia, the astroglia, the neurons, we know very well that neurons and circuitry have homeostatic responses to amyloid, and Nina Slutsky published on that. So there is a whole homeostatic response to this stress, and that takes years before it's broken. And part of the breaking of the mechanism is the tau spreading throughout the brain. That's the second hit, which is probably already, and I don't know whether the tau is really driving the neurodegeneration at that moment. It could be that the tau is part of a bigger game there, where the brain, where the neurons are collapsing. We have ourselves shown that the neurons in Alzheimer patients, and also in our mouse model, in our humanized mouse model, they start to show necroptosis markers, and the kinases are phosphorylated of the necroptosis markers. So in parallel with the tau appearance, you get also necroptosis markers which become stained, and that's also kinases. So I think that while the neurons are collapsing, a lot of kinases get activated. They phosphorylate the tau, which disconnect from the microtubuli and is then able to form tau inclusions and tangles. And they activate other kinases which induce necroptosis. And maybe there are other aspects there in the cell which we don't understand. So I think it opens a lot of interesting boxes. But I also think that that stage is already a late stage to do something to prevent the whole system. So at that moment, we need to stabilize the disease or do whatever we can to halt dementia, yeah, basically.
Maybe I have a provocative discussion to launch here. I was hit a little bit by the humanized mouse model that you proposed there, where you derive these human microglia and then reinsert them, let's say, in the mouse brain. I think back in time there has been also this speech gene that have also been looked at for that, the FOXP2, if I recall correctly. Now, we get these microglia that are human microglia. We put them in a context that is mouse neuron, astrocytes, circuit, and so on. And I understand that then we can get some relative information, but then what are the pitfalls there? What do we risk?
Zillions. I agree. Every model has its limitations, and the art is to try to understand, to take into account the limitations and then to make conclusions. So what I also think is that if you do mouse work or model work, you have also to go back to the human patient and try to see to what extent your predictions can be confirmed, but it's always limited. The functional predictions are very difficult.
But do you think that the gene...
I will, just to say, we have a control there. So we take the microglia, the human microglia, and we transplant them in again, an immune deficient mouse, but we transplant it in the brain and we look what happens. And there we see that these microglia have a very homeostatic phenotype. And we compared those microglia with microglia we derived from surgical resected specimens from humans. And we could show that there is a very strong similarity in the expression profiles we get from the human microglia transplanted in a wild-type mouse brain with the human microglia isolated from a patient. So I think it's a very good model for human microglia. It's probably one of the best we can have at the moment, because if you put human microglia in a dish, they become very activated and do not show that phenotype. So within the limitations and agreed, so there are probably human, for instance, one very nice example is we need to humanize in the mouse the CSF1, the CSF1 growth factor for microglia, which is essential to keep them going. And so the mouse CSF1 has a couple of amino acid differences with the human CSF1. And so if you transplant, we have in this mouse the CSF1 gene, we have transformed, other investigators at Regeneron have mutated those genes to humanize them so that these mice can receive human microglia. And so that tells you already that how important it is to know the difference between the human and the mouse molecular biology. And so there are probably other aspects which we will not be able to model in this model. For instance, if there is a human-specific gene, and working on that too, so we have also these transplantations of human neurons in the mouse brain, and it works remarkably well. Pierre van der Hagen also, we use that model, and then we bring in, we didn't do that experiment yet, we can bring in the human microglia and see how the human neurons interact with human microglia. And the reason to do this is that you need a three-dimensional structure of the brain. You can try to model that in dishes. It's not easy. So the only alternative I see is organoids, human brain organoids. where you would have these interactions. But there we have the problem that it's so difficult to get amyloid plaques or neuron tangles in those organoids. There are close to what we see, to the extent we were able to model this already in mouse. And so what we do with our approach is just go a little bit faster than the organoids and trying to do human modeling in the mouse model while others are developing better and better organoids.
Thanks, Bart. So we have 2 minutes before we are automatically locked. So Tara is a freshman and probably a very young scientist, and I would say she deserves to have a little bit of this question. And basically she's asking whether the difference in genetic between families more sensitive or susceptible lies on microglia differences. Bart, your quick comment.
Well, she apologized for her question, but I think this is just a brilliant question. That's the whole essence of what we try to do here. Yes, I think that that some people are completely protected against Alzheimer's disease or against the amyloid plaques and the tangles, thanks to their genetic constitution. And we want to know these resilient genes to do something, to use that knowledge to protect those who don't have this resilient gene. So in essence, this is the main question of my group to find that. And it's not only microglia. I'm sure that there are also resilient genes in other cell types. But the microglia are certainly, as the GWAS studies tell us, part of this important piece of the puzzle. Thank you very much.
Perfect. So in 30 seconds, I would like to thank, in first place, Bart for being available for this, all of the attendees for listening to these new studies and new avenues into Alzheimer's disease. And I invite all of you to keep following Neuromatch. And Bart, has been great to have you here this morning. And thank you all for for being here. Thanks, Ali, for your technical support. And happy day to all of you.
Thank you very much. Bye-bye.
Once again, bye-bye.